Yeah, I'm Will. I'm a computational biologist based in the Department of Medicine. I work up the hill, which I think is in that direction, in the John Radcliffe. And my job today is to not only get you excited about plasmids, but to get you excited about the nucleotide composition of plasmids, which I appreciate is a bit of an uphill struggle, but hopefully, if you stick with me, we'll get there. So maybe let's start with an intro, a refresher, whatever, into plasmid population genetics, right? This little black circle here is a plasmid, right? It's an extrachromosomal replicon, and it depends on the host cell to function, right? It arrives effectively with a list of demands for its host cell, right? And so different plasmid families have different demands, and that's what defines the plasmid incompatibility groups, right? Different plasmids get on with different plasmids. One thing that plasmids are really good at, right, is horizontal transfer. They can spread across species, genus, right, maybe even the phylum barrier, right? Over time, a lineage might lose a plasmid, it might find it again. And it's really that friction between the dependency on the host and the transients that drives all of those really fascinating dynamics in plasmids. So, for example, this absolute mess of genes here, right, is about 150,000 base pair conjugative plasmid, right? And I'd say it's pretty archetypical of a clinical plasmid. So we've got this large swathe of genes at the top left, right, controlling those essential functions, right, the things the plasmid needs to survive. So it's maintenance, it's transfer, it's replication. And then kind of smattered around, right, we see these little insertion events. We see this class 1 integron at the bottom left, which has facilitated this multi-drug resistance region, right? And this is kind of classic of how plasmids evolve, right? We have this conserved set of kind of backbone genes, which the plasmid needs to survive. And as they move through a bacterial population, right, they're gaining and they're losing genes. And so the way I kind of like to think about plasmid evolution is along these two axes, right? In the Y direction, we've got that kind of classical understanding, right? We've got mutations fixing in a population. But then along our x-axis, we've got this kind of more coarse-grained movement, right? This structural change where genes are coming and going. We've got rearrangements. We've got recombination events. We can also have fusion events, right, where two plasmids co-integrate, and we end up with these multi-replicon monsters, right? So I promise this will become relevant in a second, but I'm going to move on to the meat of the matter, which is plasmid GC content, right? And it's a very simple statistic. I think it's maybe a deceivingly simple statistic, right? We're just simply calculating the proportion of nucleotides, which are Gs and Cs. We're taking that entirety of our sequence and we're collapsing it to a single data point. And I do think it's really important. I mean, I think ultimately it gives fundamental insight into how plasmids are evolving, right? As I've written there, GC content can influence gene expression, it can influence DNA stability, and indeed it can be driven by myriad factors, right? Is it selection? Is it drift? Is it biased gene conversion? And these drivers are going to be acting on different levels and in different amounts. And for plasmids, it's not necessarily clear what's going on. So what I'm going to talk about today is a project which is beginning to try and peel back some of these layers. So let's build a data set. This is quite a dense slide. I appreciate that. But effectively, what we've done is we've started with this fantastic resource called PLSDB, right? What it's doing is it's pulling all of the plasmid sequences from NCBI, it's making sure they're non-redundant, and then it's applying some QC. So most of the work was done for us, right? So we've got about 60,000 to begin with. And then it's just a case of filtering out what we want and what we don't want. So we want circular plasmids, right? We want to look at the whole sequences. We want complete assemblies, because we're going to do some fiddling with the chromosomes in a minute. We want single replicons, because we don't have to deal with those messy fusion events. And then the other thing we did was filter out col-like plasmids. I'm not going to get into why I filtered them out today, or indeed get into them at all, because they're kind of a different kettle of fish. But if you are interested, please come and speak to me. But what we ended up with was 12 replicon types, right? And I would say these are mostly clinical offenders, but that's kind of the benefit or the challenge of using public sequences, right? Most of these are conjugative, and most of these crop up in outbreaks, nosocomial outbreaks in hospitals and the community and whatnot. But this is our data set. And so the first thing we did was, of course, calculate the GC content. And the first thing I think we see is this massive spread of values, right? We're going from about 39% on the left all the way up to about 54% GC content. And I know some of you in here work with mycobacterium, right? And I think that hovers around mid-60s, maybe. So these things are incredibly variable and generally on that kind of skewed side of 50. And I think the classic result, right, whenever you're reporting the GC content of plasmids, is that plasmids are AT-rich, right? If we take the GC content of the plasmid and subtract that of its chromosome, we see this massive skew to the left, right? All of these orange bars represent AT-rich plasmids compared to the few blue on the left. And we can quantify this, right? We can kind of build a mixed effects model, and we can do all that fancy statistical stuff, and we can see that on average, the plasmids in this data set are kind of 2, 3, 4% richer in AT content compared to our chromosomes. Cool, okay, so the chromosome's driving this, let's go home, right? But obviously it's not that simple. And what we find is actually, when we consider the variance, right, that a model like this explains, the chromosome is an abysmal predictor for the GC content of the plasmids, right? It's explaining about 0.3% of the variance, right? Whereas something like replicon type, right, those 12 categories earlier, is explaining upwards of what, 67, nearly 68% of the variance. And I think that really becomes clear on this next slide. As I was saying, right, the chromosome, abysmal predictor, whereas something like replicon type is driving so much more of the variance in our plasmids, right? And I think that becomes clear when you actually start to plot it out. So all I've done is I've stratified the GC content by these different plasmid families, these different plasmid lineages. And I hope what becomes clear from this plot is the great spectrum, right, of values these different plasmid replicon types can take. Some of them, right, like the ink Cs and whatnot, are covering this massive range of relative values, whereas some of them are quite tightly packed. And then we also see within these distributions little kind of subclusters, right? Little tightly dense packs of points, right? And they might be representing genuine subpopulations, they might be representing outbreak sampling, you know, that's not necessarily clear. But I think what we can take away from this, right, is the sheer diversity of GC content across these plasmids. And I think what's also important is that the within plasmid GC content is highly variable, right? Just taking this plasmid from earlier and just kind of whiting out the genes, this black line is the GC content of this plasmid. And it's not a smooth line, right? It's this incredibly jaggedy, variable distribution. And so when we're calculating our GC content, we're collapsing all of this variation to a single point. And so if we can't rely on the chromosome to tell us what's going on, I think if we want to understand what's driving this AT-richness in plasmids, we need to look within the plasmid. And that's what we did. So the first place to look, I think, was quite a natural one, and that was just to compare genic and intergenic sequence, right? So the stuff encoding proteins and the stuff containing your regulatory regions, your promoters and whatnot. So from the plasmid's perspective, when it gets into a new cell, it wants to express itself, right? It wants to maybe offer some fitness benefits some of the time. That's probably its job. So if our promoter regions are AT-rich, right, potentially that DNA is more malleable and it's easier for its machinery to initiate transcription. That seems fair, right? But from the host perspective, it wants to control that plasmid. It doesn't want it to express all the time. And so something like the H&S protein, right, is going to target those AT-rich regions like promoters. And so if some of these mechanisms at all are in play, what we might expect to see is during the intergenic regions a dip in the GC content, right? We might expect the genic regions to be richer in GC than the intergenic regions, okay? So what we did was effectively explode our data set into intergenic and genic regions. So again, these 12 facets are our 12 replicon types in the data set, and we've just stratified it by genic and intergenic sequence. And I hope what's clear from this is the massive amounts of variation, right? But again, we can try and discern what's going on statistically, and what we do indeed find is that the intergenic stuff is by far more AT-rich. So on average, our genes are clocking in at what, 2, 3, 4% AT-rich compared to the chromosome. But in our intergenic stuff is a further 4% on top of that. And so when you plot your GC of your plasmid and you're seeing all of these jaggedy lines, that variation is being driven by the intergenic sequence being more AT-rich. So cool, okay, we've got this dichotomy between genes and intergenic sequence. The next question is, what's actually going on with the genes themselves, right? And so this comes back to those ideas from earlier about this plasmid backbone moving through the population. So if a plasmid lineage is successful, what we'd expect over time is those core genes, right, the ones involved in transfer, replication, maintenance, and so on, are going to increase in frequency, right? And so what we're going to see is if we take all of the genes from that plasmid lineage, we're going to see those effectively coming out as core genes, if we want to talk in kind of a pangenome-style way. Whereas those genes that are being gained and lost, for instance, those that are moving on transposons, are going to manifest as our accessory genes, right? They're going to be very low in frequency. And so what we can do is we can take all the genes in our plasmid lineage and we can effectively collapse them into homologous clusters, right? And so for each gene, we can effectively get a frequency value in our plasmid population. So going all the way from 0 with our kind of rare accessory genes, those big explosions on the left of the facets, and then on the right are kind of less frequent, or sorry, less numerous core genes. And so the first question, right, is gene frequency involved at all with AT-richness? And the answer is, I guess, sort of, slightly. I mean, it's certainly significant, but it's also certainly minuscule, right? As a gene goes from being about 0% in the population to around 100%, it's gaining 0.15 percentage points in AT-richness. It's not worth writing home about. I think what's more interesting is the shape of that distribution, right? That kind of funnel shape you notice as they increase in frequency. And the way we kind of characterized that was by looking at the variance away from that lineage average. So for every gene in our population, we effectively average the GC content, right? And what we wanted to understand was how does the distance from that point vary as the genes increase in frequency. And so for our accessory genes, right, that big explosion on the left, they're around 7% away from that, right? There's this great massive spread in GC content for our accessory genes. But as we drift towards the right-hand side, right, when we're dealing with these backbone genes, we're seeing them only be about, 3% away from this average. So what we're seeing is this effective convergence towards this lineage-specific average. What's driving that? Well, I think that's our next question, right? Is there a strong purifying selection acting on these core genes? Potentially. Is that pushing it towards some sort of lineage optimum? Potentially, right? Or is it because a homogeneous backbone is generally more stable as it moves around a bacterial population? So where do we kind of go next? Well, we've got the result in our back pocket, right? This intergenic sequence is AT-rich. And we've got this idea that as we're increasing in frequency, we're becoming more homogeneous. Cool. Well, I think a next step to look is at the host range, right? If individual GC content of the chromosome is not telling us what's going on, perhaps we need to start looking at multiple chromosomes. These plasmids aren't stuck with one lineage, they're spreading around, right? So maybe they're aiming for some sort of average amongst them. I think the other thing we need to look at is gene function, right? So for example, insertion sequences are generally AT-rich. So if your plasmid lineage is enriched for insertion sequences, you're generally going to fall on the AT-rich side of things. Amazing. Let me scroll down. So all that is left... Oh, I can't go any further. Well, thank you, Anna. And thank you to those that have funded me. Thank you to Laurie as well, who's been brilliant. Thank you to Liam for some great conversations. And thank you also to some attendees of the EMBO Plasmid workshop in September, particularly Ed File and Eduardo Rocca for some really helpful conversations that kind of kick-started this project. Thank you very much.